Hi guys. Okay, here we go on the next chapter in biology. It's chapter 7, which is, I believe, titled Cellular Processes. And the two big ones that we are going to talk about, because they really are the most important to us, are photosynthesis and cellular respiration. So, a little bit about photosynthesis first of all. And I know I say this about a lot of things, but this was really one of the most important things that was ever discovered, ever. Because it is the first step in our our access to energy from the sun. And so I go through a little bit of the history. There are several men who played a big part in photosynthesis, which I know some of you are like, what in the world were they on? Why are they studying this so much? There are some people just like to know things. And so some of the history, first of all, the Greeks, remember the Greeks had thoughts about everything. And they thought that plants just got food from the soil. Like the sugars that we eat from plants, or, the fruit in that, back then, just was soil that had been converted. They weren't right, and it took a long time, but eventually people started doing experiments. And the first guy is this handsome fella who is named Jean van Helmont. So this is him over here. I know you want to see his beautiful, handsome face. He experimented to prove that plants obtain nutrients from water. For what he did in the experiment that he did, he wasn't quite right, but he did know that water played a part, and so that was kind of the first step. The next guy, Joseph Priestley here, another handsome feller, did different experiments to prove that plants restored impure air. And so you guys might still in your houses have house plants, and your mom or dad may have said, that helps to keep the air clean, and that is true. It does take out CO2. And it becomes part of a different product, and so it does help to clean some of those things out of the air. So that was good. And then this last guy, Jean Ingenhousz. So here he is. He did several things. He proved that Joseph Priestley was right, except that the plants do have to have sunlight for the process to happen. He also proposed the idea that plants break apart CO2, which remember is what the plants are cleaning out of the air, to form carbon and oxygen. Kind of right, but again, it needed some clarification. And then finally, he said that the carbon, so this carbon that is part of the broken apart, combined with water to make carbohydrates. Because remember, carbohydrates contain carbon, hydrogen, and oxygen. They missed some steps, but they led to more people's work. And so the next guy, and notice the picture's quite a bit different, a big time gap here, just technology caught up. But a man named C.B. Van Nijl proposed that oxygen was produced from the water and not from the CO2. So by this point, we had a little bit more ability to track specific elements in chemical reactions. And so we could see kind of where things were going. And then the last guy, he is the rock star of the photosynthesis world, Melvin Calvin. He did different experiments and proved that photosynthesis is a whole series of reactions, a whole series of chemical steps. But what we get from all of his steps and kind of the big picture is this equation that water and CO2 combine with light energy, which is what we get from the sun, to produce glucose, which is this sugar here, this first thing, some more water, and some oxygen. Okay, so it does a lot for us because the glucose, remember, is how our ATP, what our ATP needs to form and then break and release the energy. And then kind of as a byproduct, we get some oxygen, so we get to breathe. So kind of cool. For his work, Dr. Calvin won the Nobel Prize in 1961. So very, very important. Okay, so now moving on to the steps. And we could go into way more detail. I know I've said that before too, when we really get to photosynthesis, but just where we'll hit the highlights. Okay, so question 1 in chapter 7 is, how does a cell convert light energy into chemical energy? And so we've got some different ways that that energy gets to us. The light energy gets directly to green plants and algae because they can absorb it. They have chlorophyll and they have their chloroplasts. And that converts that light energy into sugar, which is chemical energy that the plant then eats and uses to get ATP formation, that kind of thing. So again, as we've said a couple of times, you're not green. For lunch, we don't go out and just lay in the parking lot and absorb sun. We have to get our energy indirectly, which means we have to eat the plants or eat animals that have eaten the plants, and then kind of indirectly get the energy, get the glucose that we need. So fun fact, a very small percentage of our oxygen comes only from trees and plants. So I'm not saying, go cut down all the forests. Obviously, they're very important still. But just be aware that actually we are more reliant on algae that is out in the ocean for the majority of our oxygen. So good thing there's lots of algae. Okay, so now with photosynthesis, a couple of structures to know. Big one is chlorophyll. And chlorophyll is the catalyst or the enzyme for photosynthesis. And this picture over here, so that is the molecular structure for chlorophyll. And notice what they call the head region is got magnesium, which is right here in the middle of that little geometric shaping. And then you've got the tail portion, which is just a carbon chain. Again, pretty simple as far as the form goes, but very important. It is made only if the plant has access or is exposed to sunlight and iron. Even though there's no iron in this system, that iron may act as another catalyst or just another, kind of intermediate step to get to the chlorophyll formation. And so, very important, it is the green pigment in the chloroplasts, and it's actually found in the grana membrane in eukaryotic cells, so in plant cells basically. And so the granas in the picture down there are labeled granum, singular versus plural, and the grana is the stacks. And so it is found in the membranes of those stacks, or of like those little coins. And there are different types of chlorophyll. There's chlorophyll A, which is more of a blue-green pigment, and it is reflecting energy from the blue-green wavelengths, which is why that's what we see, and absorbing energy from the violets and the reds. Chlorophyll B is a yellow-green, and it reflects yellow-green colors to us and absorbs the blues, violets, and reds. For the most part, chlorophyll A is active during the spring and summer months, because those are they're getting the correct wavelengths to stimulate them and give them the right energy. As we move into fall, the earth tilts away from the sun, which means the wavelengths, the colors coming in, the energy coming in change. And so chlorophyll B becomes more prevalent, and that's why we start to see the leaves changing colors. In other plants and in certain trees, those chlorophylls have even different colors, so you see the reds and more of the browns and oranges. But all of that is simply because of the wavelength that the chlorophyll can absorb and continue to use for the process of photosynthesis. So color change happens. It's been happening. If you look right now, we're in kind of late September, but if you're starting to drive around and actually start to look at the trees, you'll start to see some of the leaves are changing because they're not getting the wavelengths for chlorophyll A anymore. Okay, so here is photosynthesis. So it takes place in the grana, which is the stacks of coins, and the chlorophyll is located in the thylakoid membrane. So the thylakoids are the coins themselves. And two separate phases, two separate sets of reactions happen. Light dependent, so it needs light, and light independent does not need light. Okay, so a couple of steps, and then there's a picture, I believe, on the next page. So chlorophyll reacts with light and splits a water molecule into different parts. This water molecule can come from the air, it could come from the soil, it's in the cell. And so the water molecule gets split into hydrogen, which is used to increase the hydrogen concentration inside the thylakoid, inside the coin. Oxygen is released to the atmosphere. That's for us. That's what we get to breathe. And then electrons, they have lots of energy. They are used to actually pump more hydrogen into the thylakoid. That increases the concentration of the hydrogen. Energy is needed because you're going into a high concentration area, so you're going uphill to do that. So that's the need for energy there. Now, once you have all this hydrogen built up, it goes back out, leaves the thylakoid through a big protein that is called ATP synthase, and that ATP synthase will bond a third phosphate group onto ADP to form ATP. And here is the exciting part for some of this, is that you get some energy. And so that can be used from other things. Now, once that H is pumped through, it bonds to a molecule called NADP+. It actually has a real name, but thankfully they use the abbreviations. And it forms an NADPH. That NADPH is sometimes called an electron shuttle or an electron carrier, because it is going to also take in an electron along with, after it is bonded to that H, and carry things, which we'll talk about with the next phase. But real quick, here's this picture. So here is inside the thylakoid. So inside, this is kind of outside it, just in the chloroplast. So the sunlight comes in, it hits a little photosystem 2, that's that PS2, and right away you have this water down here breaking into oxygen. That's for us. Yay for us. We get to breathe that oxygen. And then this hydrogen. The hydrogens are getting built up, and they're also being pumped in through some of these different enzymes, which we're not going to worry about too much. So you have lots of hydrogen inside the thylakoid. So you'd have bunches of hydrogen just getting built up in here. And that takes energy, so that's where this little electron, these electrons kind of come in. And then that hydrogen will go back out of the thylakoid through the ATP synthase, which is this molecule here. And every time one passes through, an ADP gets converted into ATP. That's really cool to me. And then that H will bond with this NADPH, and this little electron will also grab a ride with that little shuttle. Okay, so really the only thing that we get out, for us personally, that we get out of this this first phase is some oxygen. The ATP, the NADPH are products, but again, we're not super aware of them, and they actually move on to the next phase. And so now we have the light independent phase. So this is in the stroma, so this is outside. So it uses the ATP that was just made, uses the NADPH, and now it brings in the CO2 from the air. So that's what we contribute, because that's what we breathe out. So the plant gathers up 3 CO2 molecules, binds them together by an enzyme that's called ribisco, again an abbreviation, and it forms a 3-carbon molecule that we call glycerate 3-phosphate. Glyceraldehyde 3-phosphate, there's several names for it, but basically we will call it G3P. Okay, so we've got the ATP from the first, the NADPH from the first phase to go along with CO2 and form G3P. So each G3P has 3 carbons in it. 2 of those get put together to make one 6-carbon glucose. Okay, so here is that chemical energy. This is kind of the whole purpose here, is to get this glucose molecule. The remaining stuff that doesn't get into the glucose, mostly phosphates, are recycled to make RUBP, which will become ribisco, which will then again join up with 3 CO2s, and you have this circle. Now, the Calvin cycle is the name of this phase, and it takes quite a bit of energy. It takes 9 ATP and 6 NADPH for the Calvin cycle to turn. And so here's a picture of that. So here up at the top, here's the CO2s coming in. There will be 3 of them, and they go through some different reactions, which we will not worry too much about those little byproducts. And 3 of them get put together to form this G3P, this glycerate 3-phosphate. And then they kind of get out of the cycle now. They get out of the circle, and 2 of them go on to form glucose. And that, again, that's what the plant stores, that's what we eat, that's what our cells will use. So it is the chemical energy that we need. Then the rest of the stuff that doesn't get into the glucose, like I said, gets recycled. So you get to this RUBP, when then really you get to this Rubisco, because it is really the thing that kind of gets it all started. Okay, so now photosynthesis. You need some things for it to happen. So first off, you need plenty of light, and the light needs to contain the right wavelengths. So that's, for the right type of chlorophyll, has to be the right intensities. You also need CO2. So again, that's what you're breathing out. So keep on breathing. And then adequate temperatures. Most plants have a pretty wide range where they function well. But if they're too hot, then certain enzymes break down. If they're too cold, then that is too close to freezing for plants. And so they're pretty consistent, or pretty, what's the word I'm looking for? They're pretty in need of kind of a narrow range of conditions. Most plants will reach a plateau where they have all of these adequate amounts of things, and then they just can't go any faster. Okay, so now what happens if things are a little bit different, if the plant doesn't get enough CO2? It was kind of the main thing. And so if they do, kind of the normal, like the process we just talked about, is generally called the C3 pathway. Because they build up these 3 carbons and then 2 get put together. But some plants don't have enough CO2 to run the Calvin cycle consistently. Usually this is plants that are in like a desert climate, because those plants have fewer openings in their leaves, which are called stomata. And that stomata is how CO2 gets into the plant. But unfortunately, it's also a big place for the plant to lose water. If it's already hot and it's already dry, the plant can't afford to lose water, and so it has fewer stomata, or it closes them. So plants have gotten a way to work around this. So first you have what they call the CAM pathway, and this is usually used by cactus. And these plants will fix, and fix in this case means to store. So they will take in CO2 during the night when it's not so hot, when they can open their stomata, not worry about losing light or losing water, and they take it in and they store it. They fix it to another molecule so it can't go anywhere, and then they go through the Calvin cycle. So same process, it just isn't happening instantaneously. And then you have what they call the C4 pathway, and this is used. You usually see this in plants that have like the narrow, kind of skinnier leaves. Corn, crabgrass, certain flowers have really, really skinny leaves. But these plants don't do anything necessarily at night, but during the day they tend to partially close their stomata, especially when it's super hot. And then they have a 4 carbon molecule, which is where the C4 name comes from, that again stores CO2, fixes the CO2, and with these special enzymes. And so it just works as the oxygen is high and the CO2 is low because of the heat. And then later on, when it's cooler and they're taking in more, or not giving off, they're giving off more oxygen or less oxygen, then they can use that stored CO2 and go through the Calvin cycle again. So it's they've worked around it. Okay, so that's how the energy gets in. So the light dependent, the light independent phases end up making glucose. And so now we either eat the plant or we eat an animal that ate the plant. But so how do we get the energy out? well, how do we get the energy that's in that sugar? And it is through a process called cellular respiration. And this is the breaking down of food or chemical energy into usable energy, ultimately in the form of ATP. So usually we are talking about glucose, but we can use fats, we can use other sugars, you can use proteins. And there are two types of respiration: aerobic, which means we need oxygen, and anaerobic, which means we don't. Oh, hang on. Okay, sorry, something weird happened there. All right, so now, what's the difference between the two? Basically, they are the same equation, just the opposite of each other. And so this is that same equation that I showed you a couple of slides ago when we were talking about just photosynthesis, but it was aimed the other way. So everything was reversed. This is cellular respiration. Here is the glucose. So here is glucose. And here is the oxygen that we are breathing in. And we get from that CO2, which we breathe out, and then a different form of energy. So you have to have all of these parts for both of them, but it just depends on if you're using them or making them. The big difference is the enzymes that are used, and we won't really get into that, but then also the structures where it takes place. Obviously, photosynthesis takes place in chloroplasts in the cell, and then cellular respiration will take place in the mitochondria, powerhouse of the cell. Okay, so cellular respiration, we've got two phases, and the first one is called glycolysis, and it actually happens outside of a mitochondria. So this is out in the cytoplasm, and it is simply breaking down a glucose into a molecule called pyruvate. And it takes energy to make energy, so you have to have an ATP and then also have to have a glucose molecule. The products are hydrogen and electrons, which will go on and help, and ATP. So we had to use a little bit of energy, but we also made some, so it kind of evens out, and we end up with two total ATP. So that's our gross product, the end result. All right, so here is a really complicated picture of that. And so I know it looks kind of confusing, but just kind of hang with me. So we're starting up here at the top, and here's our one molecule of glucose. So see the little hexagon there. You have two ATP that are going in. So those are the two that we needed. And through a reaction, you go through some kind of intermediates. You form some different sugars, and eventually you split into two. And so that's where this kind of double arrow ends up. So now we're doing the same thing, just on both pathways. And so now we have broken down our C6 back into C3. Notice this glyceraldehyde 3-phosphate should sound kind of familiar to you. And as it is breaking down, it releases actually a total of 4 ATP. But because we already used 2, then you'd have 4 minus 2. That's where we get our 2 ATP. And then really the thing that we're after for the big energy production are these 2 molecules of pyruvate. Okay, so now phase 2 is the citric acid cycle. This is sometimes referred to as the Krebs cycle. Just kind of keep that in mind, because you might hear that, because I might say it, because that's what I learned. But anyway, this happens in the mitochondria. So the pyruvate, the 2 pyruvates that were made in the step before, move into the mitochondria, and they form an acetyl enzyme A using some different enzymes, CoA enzyme, and they produce electrons and hydrogen, some more of that, some more ATP, and some CO2. So that's just the waste product that we breathe out. Now, this step doesn't require any oxygen, but for everything to keep moving, oxygen has to be kind of waiting after this step. Okay, so here is, so notice Krebs cycle, but again, you could also have the other name on there. And so here's our pyruvate. So these are the 3 carbon molecules that are coming in from glycolysis. They meet up with this CoA enzyme and form acetyl-CoA. And then this little CoA is kind of on its own little cycle, so it just kind of keeps going back and forth. But this acetyl-CoA just continues to get broken down. And first off, you've got a CO2 that is produced. That's you exhaling. But you also have some NADH. It's a little different, so it's not a pH now, it's just NADH. And some FADH. These are super important because they are going to carry hydrogen electron to the next phase. So there's the next thing. And some ATP. So from pyruvate, you get quite a few things. ATP is important as energy, but really it's the NADHs and the FADH2s because they are where we really make the most ATP. Okay, so now phase 3. Did I say this only occurred in 2 phases? It's 3 phases. You have the hydrogen electron transport system. Sometimes I think if you're looking this up, it may just be electron transport system. Again, you know, this is how I learned it. So this happens inside the folded inner membrane of the mitochondria. Lots of surface area for the reaction to move back and forth. So that's kind of the zigzaggies. And this is the big money maker. This is where the majority of energy or ATP is made. Okay, so here we go on these steps. So the first 2 steps are kind of combined because this is where those 2 carriers, the FADH2 and the NADH, release their electrons. So they give their electrons to an electron acceptor. This, some of this energy is used to move hydrogen back across the membrane to increase the hydrogen, because remember they're carrying hydrogen and also, and building up a concentration. Step 3, the electrons are released inside the mitochondria, and they will go along and help with some energy. And then the hydrogen concentration outside the membrane is high because they've been dropping off their hydrogen here. And so high outside, low inside. So hydrogen passes back through the membrane, uses an ATP synthase molecule, which should sound familiar to you. And this releases energy, the H moving through, to form an ATP by bonding it, a phosphate onto an ADP. So there's what we're after. And then the electrons from step 3 and the hydrogen that has moved in step 4 combine with oxygen to form water. So this is where the oxygen is important, because if it's not there to join with the hydrogen, too much hydrogen builds up and then the whole thing shuts down. Okay, it's one of the feedback systems. So you got to keep breathing. Okay, so here's this whole electron transport thing. So here you see your carriers, your NADH, your FADH2, and they are dropping off their hydrogen. So that's what all these little dots are up there. They're dropping off their hydrogens, and they're dropping off their electrons. And so the hydrogens are building up, and then they're being carried across the membrane, and then they will come back through, because of the high-low concentration, back through this ATP synthase, and they will form ATP. Okay, that's what we're after. Also on here, oh, here's where the oxygen is being used. So oxygen is sitting in here as you breathe it in, waiting for these hydrogens to pump in, and so it can join with the oxygen to make water. You breathe a lot of water vapor out, even if you don't see it, or your cells can use it for whatever purpose it needs. Okay, so here's the big thing with aerobic respiration. Get lots of ATP out of it comparatively. From glycolysis, that first step, you get 2 ATP. From the citric acid cycle, you get another 2 ATP. Remember, you had to use some and make some, but you get 2 total. And then from the hydrogen and electron transport and this chemosmosis, yikes, is just the movement of hydrogen is what they're talking about there. You get 32 ATP. And so It is, good grief, and for a total of 36, sorry, typo. And this doesn't sound super efficient. We get 50 to 60% of the energy that's in the glucose. What compared to most machines and the way that they use energy, like gasoline or whatever it is, our cells get much more energy percentage-wise out of an energy source than anything that we can create. So anyway, so 50% or so is what we actually use and get. The rest is lost as heat. It's said that it is unusable. It's really not, because it is helping keep your body warm. It's helping keeping your core temperature consistent, which again is part of homeostasis, which again is part of helping your cells work efficiently. It all goes together. Super, super cool. Okay, so now anaerobic respiration. Some cells are in environments where they don't have a lot of oxygen. Some don't want oxygen at all. Others can function without it if necessary. And so they sometimes, because they are still needing to produce energy, have to use what is called cellular fermentation. And so this is the breakdown of food, again, still usually glucose, without oxygen. You in general don't do this, but your muscles, some of your muscles can do this, you know, if you've been working out long enough and you're like in oxygen debt. But for the most part, this is referring to bacteria. But everything starts just like normal with glycolysis, and it breaks down 1 glucose into 2 pyruvate. And those 2 pyruvate, depending on the cell type, it can either then go through alcoholic fermentation or lactic acid fermentation. So it also kind of depends on, well, it depends on the cells. Okay, so alcoholic fermentation. So here's your pyruvate. You've got 2 of these. It's just showing 1. It's those 3 orange dots there, but this happens twice because you've got 2 pyruvates. They go through a reaction, and they need NADH, which has been generated in glycolysis, and they produce ethanol. So the only steps, notice there's no ATP listed on here. The only energy that you get from glycolysis is that first step, the one that produces the pyruvate. This is used by bacteria, by yeast. And so when you make bread and you're using yeast, the CO2 that is released up here in that reaction, that is the gas that makes the bread rise. And then this ethanol that is down here, it is an alcohol, which means it burns off very easily. And it is actually the good smell that you smell when somebody's baking bread. So, hey, thanks, bacteria. Okay, now lactic acid fermentation looks almost exactly the same as this little picture that you saw before, but now our final product is lactic acid. But starting again with 2 pyruvate, it just shows 1, those 3 orange dots there, uses the NADH, and this one can be used by bacteria fairly often. It also can be used by our muscle cells. If you're dealing with bacteria, this lactic acid becomes kind of the tart flavor in different dairy products like yogurt or cottage cheese. So it's a pretty distinctive taste, but that gives some flavor to some of the things that we eat. As far as people go, lactic acid is thought to be the cause of muscle soreness. And so as you exercise, you start to breathe faster and faster, but if you can't keep up, you can't take in enough oxygen, then everything shuts down. Glycolysis still goes on, so you're still getting some energy, but it also means that you have to get rid of the pyruvic acid. And so your cells will have to do that in order to keep glycolysis going, and they in turn will produce this lactic acid, which can build up in your cells, and that is what can make you sore. It's one of the reasons that your coaches may say to hydrate, and it just helps kind of wash out some, kind of dilutes the lactic acid. But anyway, again, no energy is produced in this final step. It only comes from glycolysis. Whew. Okay, so that's chapter 7. And as we go through it, I know there's a lot of weird words and a lot of chemistry. Just kind of keep with the general stuff, and please, as always, ask me questions in class. Do the terms, look through the book. This really is vocab, so make sure you know what's what. All right, thanks guys.